Well, hello again, everyone, and welcome to this exciting chapter of the blood physiology. Now, we did cover quite a bit about especially the white blood cells in the immune chapter. So we will be discussing what's left of the white blood cells. We're going to focus some great details on the red blood cells and the platelets. And we will discuss together in this video the process of hematopoiesis and the coagulation process, hemostasis, how it happens, the platelets, the function of platelets. So it's a very exciting chapter to go through with. If we start with just talking general terms about the blood, we'll realize that the blood is about 8% of the total body weight. Average volume about 5 liters in men, 5.5 in men, 5 in women, and it consists of 3 types of specialized cellular or subcellular elements. And you will see in a little bit why do I say subcellular. Erythrocytes, these are the red blood cells, and we'll see later on more details about it, the shape and the function of those cells. The leukocytes, leuko means white, so these are the white cells. We did talk about the B lymphocytes and the T lymphocytes in the immune chapter, in the monocytes in great details, but we will touch base on them again one more time in this chapter later on. Then of course we have the platelets. The platelets are not really cells, they are cell fragments, and they're very, very important in the process of hemostasis or hemostasis, which is the stopping of bleeding or blood loss. So we will talk about each and every one of those, and especially the platelets and the erythrocytes, because these are 2 components that we have not talked about before, unlike the leukocytes, which we covered in the immune chapter. Little characteristics, a little bit of introduction. It's sticky. It's depends whether it's oxygenated or not oxygenated. If it's oxygenated, it will be scarlet. If it's deoxygenated, then the heme group will change to dark red. pH is about 7.4. Temperature, that's the core temperature of your blood. Again, it depends where the blood is. It's about 38. And we discussed the body weight, its relation to body weight and the volume before in an earlier slide. Functions of the blood: distribution of oxygen, nutrients, metabolic wastes, hormones, and it regulates the body temperature by absorbing and distributing heat. It regulates the pH because it can buffer it. And we did agree that one of the most important buffering system we have is the bicarbonate. And but we will talk more about the buffer system we have in the blood and the proteins, the function, how they can also buffer your blood pH in later on during this presentation. And it regulates the adequate volume, and we will talk about that in another chapter called electrolyte and water balance. And you will see how important the blood component is, and we're talking about the plasma here, in maintaining adequate fluid volume. It also functions in the protection against blood loss, and that will be the hemostasis we will talk about today. And it is protection against infection, and we did talk about that in great details when we were touching the immune system and the immune physiology. The blood is considered a fluid connective tissue. That is a sort of a weird combination. How come it's a tissue and it's fluid? Well, if it has interstitial space, and that will be the plasma, and the formed elements, and these will be the erythrocytes, leukocytes, and platelets. A lot of people like to mention the blood or refer to the blood as a fluid connective tissue. where those cells are suspended. There are certain terms you need to become familiar with. One of them is the hematocrit value. And the hematocrit value essentially is the percentage of blood volume that is the red blood cells. And 47% of the blood is red blood cells in male, 42% is red blood cells in female. And you can see immediately that the male have slightly higher red blood cell content, and that's due to several factors. One of them may be the menstrual cycle, but the main important factor is the fact that male have androgens, and the testosterone mainly is contributing to the remanufacturing or the manufacturing of large amounts of red blood cells through the stimulation of a very, very important hormone we will get to learn about later on in this chapter, called erythropoietin. And so immediately here you see that the male indeed have a larger percentage of red blood cells when we compare that to the female. So if we take the blood and spin it down, you will separate the cellular compartment, and the red blood cells being the heaviest, they will go to the bottom. And in between you will have what we call buffy coat. That's where the white blood cells are, and the platelets. And then you will have the clear plasma. If you leave the blood to coagulate, and we'll talk about the coagulation process later on, and then you spin it down, then what's left here would be serum, not plasma. So essentially serum is the same as plasma, but without all the coagulation factors, because you lost those, and then you will get some of the juice from the red blood cells and the cells and the platelets, because when the clot, as we will see later, contracts, it starts to squeeze out some of that juice. But nevertheless, now the hematocrit value is the proportion of red blood cells to the total amount of blood, and that's a very important measurement because you can diagnose anemia, or you can diagnose certain disorders based on just looking at this. Okay, so we did talk about this. This is a very interesting slide, and I always encourage you to read those tables after you have studied the chapter, because they summarize about 10-20 pages of text, as you see here in this example, which I placed for you. Now let's put the cellular compartment and the subcellular compartment aside, and let's focus a little bit on the plasma. The plasma have water, proteins. The proteins will compose about 6 to 8% of the plasma total weight. It's a lot of proteins there. And we will have three main groups of plasma proteins: albumin, globulin, and fibrinogen. Fibrinogen will be a key important factor in blood clotting. Albumin is the most abundant plasma protein, whereas globulin, we have alpha, beta, and gamma globulin. Gamma globulin is essentially your main, almost all your antibodies are, or all your antibodies are immunoglobulin or gamma globulins. And then we have alpha and beta globulin, which have a lot of functions. Some of them are part of the coagulation cascade, which we will study later today. And some others function as carriers for water-insoluble molecules, such as thyroxine and also some steroids. They bind to the globulins in order for them to be moved from one place to another. Cholesterol is the same thing, and cortisol. So other components of the blood plasma, nitrogenous byproducts or nitrogenous waste. Lactic acid is not a nitrogenous product, but it's sort of a waste product from anaerobic respiration, as we studied earlier on in our biology studies. Urea, creatinine, all these guys are nitrogenous byproducts. Urea going to be excreted, or big part of it, not all of it, is going to be excreted in the urine. Now, it also carries nutrients, and will have glucose, and that's the main carbohydrate you have in the blood. We'll have amino acids, and the lipid will be carried with special molecules, and we will talk about that when we get to the metabolism chapter. Now, the blood contains a lot of electrolytes, or the plasma has lots of electrolytes: sodium, potassium, calcium, and chloride, and bicarbonate ion. It contains respiratory gases, and we talked about that when we discussed together the respiratory system: oxygen, carbon dioxide. And these are the only two that will be there, and unless you are at a very high pressure, like you are in deep water diving, and then you will have nitrogen as well. And of course we have the hormones roaming around in the blood plasma. Now, that was the plasma. We're going to get back to it when we talk about the coagulation process, because it's very, very, very important component of it. But we will discuss today now the formed elements we have. Why do we say formed elements, and why don't we say cells? Well, it's the red blood cells, although the correct term for the red blood cells is red blood corpuscles. Because why is that? Because they don't have nuclei. They don't even have organelles. So it's very hard at this point to call them cells when they are just composed of hemoglobin and some water and some little protein and very few enzymes here and there. And the same thing with the platelets. They are cell fragments. So this is really why we call the other component of the blood, other than the plasma, we call that the formed elements. Okay, and so if we take a smear of blood and spread it on a slide, a drop of blood and spread it on a slide and stain it, we will see some of the cells we are talking about. We will see the neutrophils over here, and we'll have some lymphocytes. Here we see an example of a monocyte, which will we will describe that later on. And you can see also some platelets here and there in that blood film. But the major component of the blood, as you see here, is the erythrocytes, or the red blood corpuscles, red blood cells. And we will be talking about it in great details. Now, where all these blood cells or blood fragments are coming from, well, they all originate from the same cell, from the same stem cell. And that stem cell in the red bone marrow will be committed to one of those paths to become erythrocyte, which is your red blood cell, or will be a myeloblast, which will give rise later on to basophil, eosinophil, and neutrophil, or it will be committed to a lymphoblast, which will give you the B and T lymphocytes, a monoblast, which will give you a rise to a monocyte, megakaryoblast, which will become megakaryocyte, and will give you a rise to your platelets as fragments that will come out from the megakaryocytes, as you will see during our presentation. So let's start with the biggest component, that is our erythrocytes, or red blood cells, red blood corpuscles, if you want to be technically accurate. Contains no nucleus, no organelles, no ribosomes, so it's incapable of doing anything. That's why it's very short-lived. It lived like 90 days to 100 days, 120 days if you push it. And why is that? Because it doesn't have any way to regenerate itself. Whatever it was manufactured to have, this is the stuff that's gonna last with it forever. There is no way to rejuvenate itself. And its structure is very well suited to carry oxygen. It's a biconcave disc. It's shaped as biconcave discs. That means it provides a very large surface area for the diffusion of oxygen and carbon dioxide. and it's very thin cells, and that enables the oxygen to diffuse rapidly between the exterior and the innermost regions in the cells. It's almost like a pancake flattened out. And it has a very flexible membrane, and we will see in a little bit what happens when anything wrong happens to that membrane in cases of thalassemia, for example. And that's where the red blood cells will have very hard time surviving because they rupture very easily. So this is again what we talked about, the structures that contribute to the main functions. It's biconcave. It's mainly hemoglobin, and that's if you don't count the water. If you exclude the water and count the constituents of the erythrocytes interior, you will see it's 97% of hemoglobin, and that's very, very important for them, because that's what they use to carry oxygen mainly, and of course they help in carrying carbon dioxide and nitric oxide and hydrogen ions and bicarbonate. We did study that in great details when we were talking about the respiratory system, but we will touch base on it today as well. It does not have mitochondria, so the ATP is produced by a process called glycolysis, and that's very important because what happens is these erythrocytes are really carrying oxygen, so you don't want them to nibble on the oxygen they're carrying. And so they're very well suited to the function of carrying oxygen because they have no use for it internally. They don't have mitochondria to conduct aerobic respiration. Instead, they only rely on anaerobic respiration. RBCs, or red blood corpuscles, red blood cells, are dedicated to respiratory gas transport. The hemoglobin will bind reversibly to oxygen, also with carbon dioxide, although they bind to different regions, and we will talk about that in a short bit when we describe the structure of the hemoglobin. So if we look into the structure of the hemoglobin, we'll see that it has a protein, globin, and that has two components, two alpha and two beta chains. And then we have four heme pigments that are embedded within those globin chains. The iron atom in each heme pigment can bind to one oxygen molecule. So in total, each hemoglobin molecule can bind to four, can transport 4 oxygen. Where does the carbon dioxide being then bound to or carried by? It's mainly carried by not the heme, but by the globin protein. So the carboxy hemoglobin, that refers to carbon dioxide binding the globin, not the heme itself. Okay, we did talk about the ion. Some of these slides will be redundant because I combined 2 different sets of slides. And again, it is composed of globin and heme groups. So if we look at the structure of the hemoglobin, we'll see here, here is a globin and another one and the third and the fourth, and then you will see 1, 2, 3, 4 heme groups. If we look at a closer look at the heme groups here, we will see that it has a center that is of iron, and then it has this structure, this very convoluted structure, and that's where you bind to the oxygen at the center of the heme group. And because of this, you change the color of the heme from dark red to bright red. Okay. So the primary role of hemoglobin, we all know it carries oxygen. Carbon dioxide is mainly carried by the blood in the form of bicarbonate ion, and so it's not, I mean, it does carry carbon dioxide, but that's not really the main function. The main function is oxygen. However, it does combine itself with those following gases: carbon dioxide, also hydrogen ion portion of the carbonic acid. We did talk about that early on when we were discussing the process of of respiration, and that's a very important thing because it can share in buffering your blood. So in addition to the proteins that are present in the blood, the ability of the hemoglobin to bind and quenching or sequestering the hydrogen ions away from your blood enables you to buffer your blood to physiological levels. It carries carbon monoxide, although of course that's not a natural thing, and that's a problem if it binds, because it binds to the heme group with a much higher affinity than oxygen, and that can of course result in carbon monoxide poisoning, as we all know. Hemoglobin can also carry nitric oxide and distribute it around, as we have studied earlier on. The process of making red blood cells is called erythropoiesis. Poiesis is the making, and erythro is the red blood cells or the blood cells, and so erythrocyte is the red blood cell. Erythropoiesis is the making of the red blood cells. Okay. Red blood cells will survive about 100-120 days, and we agreed it doesn't have Golgi, doesn't have mitochondria, doesn't have even ribosomes, so whatever they're created with, that's supposed to last them their lifetime, which is about 100-120 days. They must be replaced regularly at the rate of 2 to 3 million cells per second. And the graveyard for the old red blood cells will be mainly the spleen, because of the tighter space. They will get into very tight space, and when they are circulating through the spleen, and that's when they start to break up. And but you can't just allow them to break up. Hemoglobin is a very important molecule, but it's only important when it's inside a cellular structure, inside a membrane, inside a red blood cell. And you can't allow free hemoglobin to roam around inside your plasma, because that can cause destruction of many things. It can cause destruction to the heme group, can cause destruction to the brain, can cause destruction to your kidneys especially. So whenever you have hemolytic anemia, or whenever you have adverse reaction to blood transfusion, and we will talk about blood transfusion later on today, it's a very big problem when hemoglobin starts to come out from the red blood cells, and will eventually precipitate in the kidneys, and that can destroy the glomeruli, and that's one of the main causes of death, as a complication of mismatched blood transfusion. In any event, the spleen will remove the old erythrocytes out of the circulation, and you will see what happens to the hemoglobin that gets released in the next couple of slides. Erythropoiesis, which is the generation of red blood cells, that's what we are talking about now, will start in the red bone marrow, not just any bone marrow, in the red bone marrow. The pluripotent stem cells will differentiate into the cells that will be committed to give you the red blood cells. As you see here, here is the pluripotent stem cell, and if it gets committed to become pro-erythroblast, and the pro-erythroblast will become early erythroblast, and then late erythroblast, and you will see that the nucleus is being spit out, then it becomes a reticulocyte when you spit out the nucleus, as you can see, and then all the organelles inside will start breaking apart until you get into the biconcave shape that we are all familiar with the erythrocyte. So it enters phases after the commitment into a ribosome synthesis, and then hemoglobin accumulation, and then ejection of the nucleus to finally become your erythrocyte. Okay, so if we have too little or too few number of RBCs, that will lead to tissue hypoxia. If you have hypoxia in the kidney, then it will sense the problem, and it will start synthesizing a hormone called erythropoietin, and the erythropoietin will go to the red blood cell, and the red blood cell will start making, will go to the red bone marrow, and the red bone marrow will start making much more red blood cells. So now we have, of course, recombinant or synthetic, we can't call it synthetic, but it's a recombinant. It's made outside the body, erythropoietin, and they are using this to treat a lot of disorders, and it's becoming the number one money making in the market nowadays, when we're talking about recombinant drugs, I mean. So we have to have a balance between the production and the destruction, and that will depend on the control and the adequate supply. Do we have enough iron to make hemoglobin? Because we can, you know, what's the point of making red blood cells when we don't have the hemoglobin inside them? So of course we know that iron deficiency anemia is a serious problem, and the reason for it is that you don't have enough hemoglobin. Also, if you don't have enough proteins to make the globin portion, that can lead into a problem as well in the synthesis of red blood cells. Vitamin B, and mainly vitamin B12, the absence of it, of vitamin B12, can contribute to a kind of anemia called pernicious anemia, and we will talk about that in details in a few slides. Erythropoietin is the hormone from the kidney, and that will directly stimulate the erythropoiesis, and that's in response to hypoxia. So of course, as we discussed together during lectures, that is both physiologically important for people, for example, who are living in for a long period of time in high altitude, where the partial oxygen pressure is lower than normal, and therefore the kidney will be in almost a constant state of hypoxia, and therefore you will see their blood cells being thicker. Why is that? Because you need more red blood cells. And the same thing if someone has a COPD for a longer time. Of course, you would expect their hematocrit value to be thicker, because you want to make more red blood cells to carry whatever available oxygen out there. But athletes, some have been using this phenomenon of regeneration, of making new blood cells, or the process we termed erythropoiesis, in what we call blood doping. And in blood doping, they would donate their blood to themselves, and they keep the blood in safe keeps for later on. And because of the fact that they took some portion of their own blood, the erythropoietin will start to make new red blood cells, and then they inject back the blood that they stored for a week or so back into their system, and then now you have doubled the red blood cells, thinking that this will help them, giving them an advantage, especially with marathon runners or cyclists, giving them the advantage that now they have more red blood cells to carry the oxygen and the carbon dioxide than the average people. But of course that increases the viscosity, the thickness of the blood, and that contributes to really nasty problems that can occur in these athletes that are using this method called blood doping. Okay. So erythropoietin will cause rapid maturation of the committed red bone marrow cells. It will increase the circulating reticulocytes, and then thereafter the red blood cells in 1 or 2 days. The testosterone also, as we mentioned earlier in this presentation, it directly increases the erythropoietin production, and that's one of the reasons we have a slightly higher RBC count in male, and that contributes to the earlier values we discussed of the males having higher hematocrit value than that in the females. So here's a picture of the erythropoiesis principle. When you have a lower oxygen amount being carried by the blood, that will stimulate the kidney will give erythropoietin. Erythropoietin will go to the red bone marrow to stimulate the production of the red blood cells. What do we need for erythropoiesis? We need nutrients, amino acids, lipids, carbs, and the lipids, of course, will make the phospholipids, that is the bilayer. It still is a cell. It has the normal cell membrane as any other cell. Iron, it has to be stored as hemoglobin. We don't allow really the iron to roam around. It has to be captured by something. And you will see once we start degrading the red blood cells, we have to take that iron, quench it away, or else it's gonna cause troubles if it starts to precipitate in areas like the pancreas or the brain or the kidneys. We can't allow that to happen. Vitamin B12 and folic acids, they are very necessary to DNA synthesis and cell divisions, and the lack of vitamin B12 can contribute, as you will see in few slides, to what we call pernicious anemia. Okay, so the lifespan, we agreed, of the red blood cells, about 100 to 120 cells, 220 days. The red blood cells will become fragile, and the hemoglobin will become to degenerate. The macrophages will engulf the red blood cells in the spleen. Now what happens to the cell, but the heme and the globin, or what makes the hemoglobin now, is very important for us, how to salvage that. We can't allow iron to again roam around, so we salvage it for reuse. We take the heme, which will be degraded to make a yellow pigment. Immediately will be uptake to make this yellow pigment we call bilirubin or bilirubinogen, and the liver will secrete the bilirubin into the intestine. Bilirubinogen that comes out in the urine, and then it will become bilirubin once it's exposed to oxygen. That's a whole different story. We'll talk maybe about this topic when we get to the kidney. Degraded pigments will leave the body, but very little of that. But most of the iron that we will lose, we can recycle them. The globin, however, is metabolized to become amino acids. So here's what happened. Hemoglobin again will break into heme and globin. A heme will be either quenched and taken into making this yellow pigment we call bilirubin or bilirubinogen, and that will go to be excreted in the liver or in the kidneys. Or you can quench that heme and you can store it as ferritin or hemosiderin, and then you can use that later on again for the remake of red blood cells. The globin, on the other hand, it will be broken into amino acids, and the amino acids you can start using them over again. Even though the bilirubin will come out from the kidneys, in the presence of vitamin B12 and folic acids, then you will start, or in presence of intrinsic factor, then you will start reabsorbing that iron from the intestine, and you can recirculate that iron back on. Things can go wrong in this homeostasis of the red blood cells. With erythrocyte disorders, we can have anemia, where the blood has abnormally low oxygen carrying capacity. The reason for the anemia can be variable. We can have insufficient erythrocytes, whether that is because of hemorrhage or because they're breaking up very quickly. We call that hemolytic anemia. That is true in case of sickle cell anemia, in thalassemia major and minor, in blood transfusion, in some poisons, snake poisons that cause hemolysis of the red blood cells. At any event, we have red blood cells rupturing prematurely before their 100 days are due, and therefore, of course, you have insufficient erythrocytes. Aplastic anemia is the process where you do not make enough red blood cells, and that can probably, the problem is the red bone marrow, a person being subjected to radiation or chemotherapy, which in turn affects the stem cells, and now you're not making enough red blood cells as you should. Maybe you don't have erythropoietin, like in cases with kidney failure. You're not making enough erythropoietin, and therefore you're not stimulating the production of red blood cells. So all of these will contribute to a kind of anemia that is the main reason for it is insufficient erythrocytes. Now you can have low hemoglobin, whether it's protein part or the iron part, which mainly is the iron part. Iron deficiency anemia, it can result either secondary from hemorrhage, or you're not taking enough iron containing food, or you have impaired iron absorption, like when you have the intrinsic factor, which is supposed to be made in the stomach. If you don't have that intrinsic factor, which binds to the iron and allows its absorption, if you do not have that, then of course you have a problem. Then you have iron deficiency anemia. Pernicious anemia is deficiency of vitamin B12, and we agreed that the vitamin B12 is very important for the making of the DNA, and if you don't have that, then of course it's very hard to synthesize great numbers of red blood cells every minute and every day, and therefore you will have anemia. And of course you treat that with giving vitamin B12 supplements. Abnormal hemoglobin, in cases like thalassemia, where you have faulty globin chain, and it depends whether it's the alpha, the beta, or both of them, and whether it will become thalassemia major or thalassemia minor. And the red blood cells in this case will become very thin and delicate, and they break up very, very easily. And then we have the more common one, which is sickle cell anemia. There's a defective gene that codes for abnormal hemoglobin, and that causes the red blood cells to be elongated and fairly rough. And when they tumble, I think I have a picture here. So this is a picture of, it's a scanning electron microscopy picture of a sickle cell. And you can see how a sickle cell becomes elongated, and that makes it very hard to squeeze by in organs and very easy to rupture, and that is a heritable disease, as we all learned about this problem when we studied biology. Okay, that was the anemia, too little red blood cells. But we can also have too much red blood cells, and we call that polycythemia. Polycythemia. Now the problem, why do we have too much red blood cells? Is the problem in the red bone marrow? Do we have a tumor and that causes the red blood cells to be generated at a much higher level? If so, we will call that primary polycythemia, or polycythemia vera, and it's a tumor-like condition of the bone marrow making too much erythropoiesis, and that will contribute to the high number you see. Why is that a problem if you have too much red blood cells? When some of you will say, well, the more the merrier, why should we care if we have too many red blood cells in the blood? Well, that will contribute to the viscosity. It will be, your blood will be thicker, slower to move around, and more prone to coagulation because it's slower, more problem to spontaneous blood clots. And that, of course, is a major problem that can lead to thrombosis, embolism, and strokes. So, again, the polycythemia here is more red blood cells than normal. One of the reasons can be the primary polycythemia. The second reason is it's secondary to something else. It could be that we have too much erythropoietin, and whatever the reason is, or you have a problem with COPD, or you're living in high altitude. Let's talk a little bit about the blood typing. Those are proteins that are on the cell membrane of the red blood cells. Let's agree to this as a rule. If you have the protein, you don't have the antibody for it. That's our rule, because your own system, as we learned in the immune physiology, should not make an antibody against something that's existing in your body since birth. So if you have A and B, you will not have the antibodies against those. You will have O blood. You will have the AB blood type, which means that you have both the antigens, which is the A and the B, but you don't have the antibodies. Therefore, your blood type is AB. If you don't have the A or the B, if you have neither of the antigens, then you have the antibodies against both A and B, and therefore your blood type is O, and that's because it's 0 antigens are on your erythrocytes. If you have a type A, then the protein is A and you will have the anti-B. Remember, you will have the antibody against the proteins that your system don't have. So if you have A, you will have anti-B. If you have B, you will have anti-A. If you have A and B, you can't have any of them because that's your own protein, and you can't build immune system against your own proteins. And does it mean that we can be exposed to the wrong blood once? Let's say that a person has blood type O. Can he take for the first time any blood? Doesn't matter if it's an A or a B or O? No, because even though that these proteins do not exist in your system, later on in life, a lot of bacteria have proteins that resemble the A and the B proteins, and that results in the stimulation of the immune system against the A and the B proteins, even though you had no prior exposure to A and B proteins. So your immune system is already on the lookout for A and B just because you have had exposure to bacteria that has proteins that resemble, and they're everywhere, that resemble A and B proteins. Whereas if the person is already AB, he cannot synthesize any antibodies against A or B, even if he is exposed to those bacteria, because his immune system should not recognize A or B protein, because they are part of your own portfolio since you were born. Okay? So here what happens if you have a donor blood with a B type, and you give it to a person which has an A type blood, and then you will see the antigens and antibodies are reacting together, and that leads to either hemolysis or clumping of the blood and thrombosis. And that, of course, happens a lot, not anymore a lot, but happens in mismatched blood transfusion. This used to be a problem in the past when people didn't find out about the type of blood before they give it, like in wars and stuff. They weren't that advanced as we are now. But nowadays, it's only if it's an accident, if a blood is mislabeled or something, then you start to see this kind of reaction happening. In transfusing the blood, you can transfuse the whole blood, or you can transfuse only plasma or red blood cells or platelets for people who have less platelets than normal. The presence of these proteins will determine what we call the blood groups, the A and the B and the O. We did discuss that together. And in addition to them, we have 30 other types of proteins that can or may not be present on the surface of the red blood cell. The most important of those were the A and the ABO system, but in addition we have the rhesus factor, or RH for short, and the importance of that other RH factor, we will talk about that in a couple of slides from here. So again, we did talk about the A and the B and the AB and O, and we did talk about the presence of anti-A if you don't have the A protein, anti-B if you don't have the B protein, anti-AB if you don't have neither A or B. We did talk about that when we had the previous slide. So here's again another table that will summarize what we talked about with the blood types. That gets us to the rhesus factor. And the RH group, the interesting part about the RH group is the fact that you don't have prior immunity against RH, even if you are RH negative. So that allows you to receive blood that's RH positive only once. And then the second time you take RH blood that is positive, it can lead to serious problems, because then you will start to react against a protein that your body has now prior knowledge of. Similarly, for pregnant women, if their first baby is RH positive, if they are the women themselves are RH negative, then with the first baby, the mother during labor will start making antibodies against that RH positive red blood cells, and that's not going to harm the first child because he's already born. But then the second child, or the third, if he is also or she is RH positive, now the antibodies in the mother's blood will start attacking those red blood cells, causing a problem called erythrocytosis fetalis, and that erythroblastosis fetalis, and that is where the mother's antibodies against the RH will start attacking the red blood cells of the baby because of prior exposure to the RH in the blood from the first pregnancy. So that is one of the major reasons we really focus on the RH and how it became really important, because like I said, the RH antigen you can be exposed to once, but the second exposure can be fatal, and that is true if you are taking blood transfusion. That's also true for pregnant women, and which can cause the disease which I told you about, called erythroblastosis fetalis. For erythroblastosis fetalis, the mother has to be RH negative, and the baby has to be RH positive in the first occurrence, and then the second occurrence also has to be RH positive. Then every RH positive baby from that woman will die as a consequence of having antibodies. But of course you can treat those, you can desensitize the system, you can do blood transfusion even to the baby when he's inside the womb. You can reduce this immune reaction, but of course it's a very unfavorable situation. Okay, and we did cover the erythroblastosis fetalis, so we don't necessarily have to talk about it any longer. The transfusion reaction, it happens if the blood is mismatched, and what can happen as a consequence is that the donor cells will be attacked by the recipient plasma, and they will agglutinate, mean attracting them with immunoglobulins, lump the red blood cells together. When you lump them, you can clog small red blood blood vessels, and causing thrombosis all over, disseminated thrombosis. It can also rupture and free the hemoglobin, and that is a problem, especially for the kidney, and that results in renal failure. And of course, now you're getting rid of a lot of your oxygen carrying capacity of the blood, and that can result in acute hypoxia. But really, the fatal consequence of releasing hemoglobin into your plasma is the precipitation of this hemoglobin in the kidney tubules and resulting in kidney failure. How do you test for blood types? Here is the red blood cells, how they should be in a drop of blood. If I would put an anti-A on and type AB blood, it will agglutinate, and also with the type with anti-B, it will agglutinate, because remember, the AB have both A and the B. And we did talk about that earlier on, but I'm repeating myself again. If the person is type A, it means anti-A will react to these cells, so it will agglutinate, whereas anti-B will not agglutinate the red blood cells. And so you can differentiate between whether the person has AB type, A type, B type, or O type, where neither the antibodies will agglutinate. And of course, with this very simple procedure, now we know how we distinguish between the different blood types, and that helps us quite a lot in preventing and protecting people against the adverse reactions of mismatched blood transfusion. All right? That was the erythrocytes, a very exciting component of our chapter, and the red blood cells, how they're made, what are their functions, what happen if things go wrong, whether it's in transfusion or in the synthesis. You have too little synthesis, too much synthesis. We did talk about all that, the importance of hemoglobin, the structure of the hemoglobin, the recycling of the hemoglobin. What do we do with the iron once it gets away? How do we recycle? How do we capture? How do we prevent damage from the iron to tissues? A problem called hemosiderosis. No, not hemosiderosis. Sorry, that's copper precipitation. So now we will talk about the other problems that we can have. Not the problems, but the other components that we have in the blood, which are the white blood cells. We're going to start with that, and then we will talk about the platelets and the importance of coagulation. Now, the white blood cells are less than 1% of your total blood volume, and we did talk about the fact that the neutrophils leave the capillaries by diapedesis, and the macrophages also reach the tissues by escaping the blood into the monocytes, escape the blood and go to the tissues to become macrophages. We will talk about the different types and the fact that those white blood cells, they can be attracted. We learned in the immune system how the chemotaxis happen, how you can stimulate the production with colony stimulating factor, how interleukins are very important in attracting more and more macrophages to the area, or more neutrophils to the area. We did talk about that in great details when we covered together the immune system. But now we will talk in general about those cell types, and here are the distributions of them. The largest of them is the neutrophils, and that is followed by the lymphocytes, and that will be followed by the monocytes, and then finally the eosinophils and the basophils. How do you remember them? Well, you can, if you remember, the following saying is, never let monkeys eat banana. So N is the largest, that's never. Let lymphocytes, monkeys eat banana. These are the distribution of those, which one is the most common and which one is not. You can classify them based on their shape, whether the cells have granules inside them under the microscope, and these will be your granulocytes, and agranulocytes are the cells that lack this granular formation. Granulocytes will be your neutrophils, eosinophils, and basophils. We did talk about them when we covered the immune system. And the cytoplasmic granules will stain whether basic or acidic, or with eosine so they are either basic and then they will stain, we call that basophil. So these guys will look blue and these guys will look red. Eosinophil will look red and the basophil will look blue, and that is when we stain them with the right stain. They are larger cells and much shorter lived than the red blood cells, all of them, and they have lobed nuclei, and they are phagocytic cells. So these are not all the granulocytes are phagocytic, just the neutrophils are phagocytic, so I apologize for the mistake here. Okay, again, the white blood cells, we did talk about that when we covered the immune system, so I'm not gonna talk about the function of the white blood cells. We covered it in really great details, and here again are the different types of cells. Let's have a look at them, how they look like. Here are the neutrophils, and we know it by the lobulated nuclei here, and that is the most predominant cell. The eosinophil is the one that's stained red with the right reaction, and here's the basophil. You can even hardly see the nuclei here. The monocytes and then the lymphocytes, and we did talk about the erythrocytes, although we didn't yet talk about the platelets as of yet. They originate all from the same pluripotent cells, and it will become committed as we learned earlier on, and the granulocytes and the monocytes are produced in the bone marrow, and they become committed to one lineage or another, and they become matured in the lymphoid tissues, as we learned about the T cells and the B cells, how they become mature and they multiply in the lymphoid tissues. If we start with the neutrophils, which we covered in great details when we talked about the immune system, they are the specialist when it comes to phagocytosis. And you saw the video earlier in my earlier presentation where they can chase bacteria down by chemotaxis. They're attracted to them based on certain pattern, as we learned in the innate immunity, and they can destroy bacteria by process of phagocytosis, and they do other things as well. They are the first defenders. They're very important in the inflammatory response, and they clean up even dead tissues and stuff. And they're the most abundant ones, and they're the first responders because of the colony stimulating factor. Within a couple of hours of acute inflammation, significant inflammation, you will start to have neutrophils in the blood much higher than normal because of the stimulation of their synthesis that occurs as a consequence of the colony stimulating factors. Those the neutrophils, which we covered in details when we talked about the immune system. The eosinophils, they will be increased in the circulation in an allergic condition, or if we have internal parasitic infestation. So originally they are designed to destroy worms, and now that the fact that we don't have that many worms infesting our system anymore, it seems like the eosinophils now are becoming more sensitized to second best option, which would be allergens, pollens and stuff in the air, and that
they discuss now as a reason for the emerging problem of allergy, that our body simply doesn't have the natural prey to attack, so they start, our own eosinophils start to attack other cells. And remember, those were the red cells that their nuclei were like divided into two, as you saw earlier in my presentation. So the basophils, they are the least numerous and very poorly understood cells. We don't necessarily know what they are. However, we know that they secrete histamine and heparin. We think that heparin is contributing to the fluidity of the blood, because we can use heparin to prevent blood clotting. And so that leads us to maybe think that the function of the basophil is to secrete that heparin, which keeps the blood from coagulation. Histamine, on the other hand, that's not something you really want to be released in great amount, but it happens because of activated complement, or because of IgE binding to the basophils, and that can release histamine. And we all know what histamine does when we studied the innate immune system, or innate response. Causes vasodilation and hyperemia, and that causes also edema in the tissue. And if that edema is in the lung, for example, then the person is having very hard time breathing. Histamine also can contribute to the contraction of the smooth muscles, and that the rise of asthmatic attack in response to exposure to allergy. So these are the basophils. Luckily, they're not that many. They do have a function, but the main good function of them comes from the heparin, and the bad function will be, especially in allergy, will be attributed to histamine. And as you see here, those two, we learned about them before, the mast cells, if you remember. The mast cells are very similar to the basophils, but they are embedded inside the tissues, and they also give you rise to histamine and heparin inside the tissue. Okay. Monocytes, they will emerge from the bone marrow, and again, all the red bone marrow, all the blood cells and the blood fragments will be arising from the same place, the red bone marrow. When they mature, they will start to migrate and go to the tissue, when they will reside there as macrophages, and they can live for quite a bit there, from several months to even years inside. And they are phagocytes. They are the antigen presenting cells, which we studied together in the innate immune system, and they can do phagocytosis. We discussed that Alright, so these were the monocytes, or the precursors for our macrophages, and we did talk about the macrophages in really big details when it came to our immune response. The lymphocytes, I think you're all sick and tired of talking about them, the B lymphocytes and the T lymphocytes, and we do know their functions in the immune system. B lymphocytes, they mature to become the plasma cells. The plasma cells are the ones that are secreting the antibodies, the specific antibodies that they are trained to make. And the T lymphocytes, we learned that we have three different types of T lymphocytes. The cytotoxic T cells, the ones that have a CD8 on them, and the T helper, the one that has a CD4, and the Treg, the regulatory T cells that are very important in coordinating and managing your immune response, so you don't overshoot or undershoot. We did talk about them, so we're not gonna bring them up again in our blood, but just enough to remember them and talk here a little bit about their lifespan, about 100 to 300 days. The plasma cells, once they mature, they don't live that long. It's only a few days, because you remember they're producing massive amount of antibodies, and that doesn't make them last forever afterwards. Okay, so here are the different type of cells again. The red blood cells, we did talk about that. The neutrophils, and I encourage you to really study this slide here, because it summarizes a lot of the things, the description of the cells, and what do they do, and their number as well. The eosinophils and the basophils, these are the guys with the histamine and heparin granules. And we talked about the lymphocytes and the monocytes. The monocytes are the ones that will become macrophages later on inside the tissues. It leaves us only with the platelets, and we will talk about that now. And sometimes we refer to the platelets as thrombocytes. Thrombo comes from thrombus, which is coagulation. And the cytes, the cells, they're not really cells, they're cell fragments. The megakaryocytes are the original cells, and they sit inside the bone marrow, and they peak themselves through the canaliculi and then to the bloodstream, not the canaliculi, but to the bloodstream, and they shoot fragments of their own cell into the bloodstream. Each megakaryocyte, I think, shoots about 400 platelets into the bloodstream. They don't have nuclei, but they have the organelles, and that allows them to make proteins. These organelles have enzymes, and they can use they can break up ATP, for example, to make ADP, adenosine diphosphate, and you will realize how important is the adenosine diphosphate as we go in. They have high concentration of actin and myosin, so by default it should make you immediately think that, well, if they have actin and myosin, that sounds like our muscles having actin and myosin, so there must be some kind of contraction happening in. And yes, you're correct. Once they are stimulated, the actin and the myosin slide over each other, causing changes in the shape and the contraction of those cells. And we'll talk about that when we get into the aggregation of the platelets and the activation of the platelets. The platelets will remain active for about 10 days, and they are removed once they start dying by the macrophages. So here is a picture of the megakaryocyte, and you can start shedding from the megakaryocyte pieces, as you can see here, tiny little pieces of platelets, and those are again portions of the megakaryocyte being, budding out and shedded away to form those platelets. They can be stimulated by something called thrombopoietin, and the thrombopoietin is a hormone that's produced by the liver and increases the number of the megakaryocytes, and as a consequence you can increase the platelet production. If you don't have enough platelets, then you have something called thrombocytopenia, and in thrombocytopenia you have spontaneous hemorrhage, especially from the capillaries. And if you look especially on your arm, you will see small patches of subcutaneous hemorrhage. We call that pectinate hemorrhage. And that's because of the fact that your capillaries are unable to deploy the platelets to stop those minute hemorrhages that can happen from the small rupture, very, very tiny rupture, microscopic rupture of the capillaries, resulting in this kind of pectinate hemorrhage under your skin. So you need to maintain the number of your platelets in the blood. Just because they're the smallest, it doesn't mean that they are the least important. Actually, they perform a superb function in preventing blood loss and in a process called hemostasis, as we will describe now in this. So the process of hemostasis is to prevent blood loss from broken blood vessels. You can involve 3 steps. Immediately, the immediate step will be vascular spasm that's sympathetic or from the pain nerve fibers. They will cause vascular spasm, and that is the function is to limit the blood loss from that region. Then you will have the formation of the platelet plug,
How does that happen? Platelets are not supposed to bind to the endothelium, but the minute you have an exposed collagen, they will bind to it and they will start aggregating and getting activated on it, as you will see in the next slide. And then finally you have the blood coagulation, which is a process, a very lengthy process. Lucky for you, you don't have to remember all the steps in this process, just few things of that process we will talk about. But this is the steps of, the major steps of hemostasis, vascular spasm, formation of platelet plug, and then finally blood coagulation. So let's talk about the platelet, the platelets first, how they aggregate. So here is a damage. The collagen becomes exposed, and because the collagen is exposed now, these platelets will start aggregating here, and we'll talk about that in the next slide. And when they do so, you see how they change their shape. They originally were these round, perfect ones, but then they change their shape. They become star-like or spiky, and that's because of the contraction of the actin and the myosin filaments over each other. And that makes them even aggregate more, because now they're not smooth, they're not like balls, but now they're like blocks or spiky blocks, and it will cause them to aggregate even further. And we will talk about that, how they are attracted to the site and attracted to each other, and they keep building on this in something we call positive feedback, and that is due to the released adenosine diphosphate that they release, and that will activate other cells to attract more and secrete more adenosine diphosphate, and so on and so on. And that is an example of what we call positive feedback. And you will ask yourself the question, well, if they aggregate in this area, what stops them from, you know, spreading around to fill the entire capillary or artery? Well, because of the normal endothelium secreting nitric oxide and prostacyclin, and that will repel the platelets from aggregating, and keeping only the platelets to aggregate only at the damaged area. So prostacyclin and nitric oxide, these are products from the normal endothelium, and it's really meant to repel and prevent the aggregation of the platelets to this normal part, whereas they only aggregate to the damaged part. That's a very important part to consider and to remember here. In more details, we have a factor that is made by the platelets. It's called von Willebrand, and or VWF. You don't have to remember the long name, but VWF will adhere to the exposed collagen, and therefore you will start to release adenosine diphosphate because of the breaking of the adenosine triphosphate, and ADP will attract more and more platelets, and the platelets, as I said earlier, will change their shape because of the contraction of the microfilaments, and thromboxane, which is also a product from the platelets, it will stimulate further aggregation of the platelets, and when you have further aggregation of the platelets, you will have even more ADP, and that is an example of the positive feedback we have. Platelet aggregation is great, but it's not enough to stop bleeding from a big cut. You need something more. You need to make the blood clot, which becomes later on the scab that you have over the wound. Platelets are great to stop bleeding from minute problems, but it also starts the ball rolling for the blood clot formation. So, as I said earlier, if you have a problem called thrombocytopenia, you will start to have tiny little hemorrhages under your skin. You might have a slightly hindered blood clot formation, but it's not as nasty as hemophilia. So we will talk about hemophilia in great details, but I just wanted you at this point to realize and understand the fact that platelets can plug small holes. They are incapable of stopping big amount of bleeding, but you will see also that many of the factors that become secreted by the aggregated platelets they are major contributing factors to the blood clot formation. So platelets, just because they are the smallest component of your blood, it doesn't by any means that you consider the platelets become the least significant. Actually, every now and then nowadays we are discovering more and more functions for the platelets in secreting growth factors and other things that haven't been previously known about the platelets. So in order for me to make the clot, I need ropes. I need things to tie cells around and, you know, gap the wound and first start to trap the cells and tie them around in what we call blood clot. But this blood clot has to be so strong and so stretchy, and with a protein called fibrin. The precursor of fibrin is present in the blood called fibrinogen, but once I activate it into fibrin, and that fibrin will be the rope that will be tying your cells together. As you can see here, these are fibrin threads, and this is a scanning electron microscopy picture. So the blood clotting, it will involve 12 different plasma clotting factors, and they discovered that some of those are like calcium and some are repetitious. But nevertheless, it's several steps, and those several steps, without going to extreme details, they can be activated by something from within the blood. And if it's from within the blood cells themselves or the platelets themselves, we'll call that intrinsic pathway. And that's a little bit longer step, so it will take 7 different steps in order for the blood clot to be completely formed. And it's set off by factor 12, which is the Hegeman factor that's activated from becoming exposed to collagen, and this is coming from the platelets. Factor 12, Hegeman factor, is coming from the platelet, and that will start the ball rolling. And if you don't have the collagen, it will bind also to the glass in glass test tube or the plastic in plastic test tube, and that will also start the ball running by the fact that the platelets will become adherent to it, and they will give you factor 12, and factor 12 will start, the whole process of activation of the clotting cascade. Now, this is the intrinsic pathway. As you can see, it's lengthy. It takes 7 steps, but the intrinsic pathway. But then we have also the extrinsic pathway. That means the tissue now is being involved. Something outside the blood is being involved, and it's the tissue. Damage in the tissue, damage inside the tissue will start causing the blood to coagulate. That's why the more massive the damage in the tissue is, the faster the coagulation or process occurs. And we will see that in the next slide, and we will talk about the importance of thromboplastin and the prothrombin and thrombin. This is very, very important. You can't ignore knowing this guy here, the prothrombin and to thrombin conversion. Neither that you should really ignore, at least just this part. So let's talk about the lengthy process of intrinsic pathway. You don't necessarily have to know the details here, but you know that when you have damaged vessels, then the inactive factor 12 will become activated factor 12, and that will start the ball rolling. You will see in many steps here the importance of calcium. So in the absence of calcium, then you don't really have coagulation occurring, and so it's no wonder that we add EDTA or EGTA to the blood to prevent it from coagulating in many laboratory or blood preservation processes. You will see that the purple tubes that you are collecting the blood in can have EDTA or EGTA in them to prevent the blood from coagulating. Anyhow, this is the intrinsic mechanism. As you can see, it's fairly long, fairly long, the intrinsic mechanism. The extrinsic, if you have extensive tissue damage, then you will give tissue thromboplastin, which is factor 3. That will directly activate factor 10 from the inactive form to the active form, and factor 10 directly will activate prothrombin to thrombin. Thrombin is such an important factor because it plays on many things. Thrombin can activate its own release by activating factor 10. Thrombin can also activate can activate the conversion directly, activates the conversion from fibrinogen, from fibrin to the stable form of fibrin, a very hard rope kind of fibrin, and at the same time thrombin can help in the conversion from fibrinogen into fibrin. So you can see how we have two pathways. One of them is the intrinsic pathway that does not involve tissue damage, and it takes much longer time to happen, and we have the extrinsic pathway where tissue damage is involved. Okay, so here's a little summary. If we have tissue damage, then we will have, whether or not we will have tissue damage, this is actually the intrinsic mechanism, then we will have blood clot formation. But at the same time, when the Hageman factor is causing this cascade of reactions to cause the blood formation, at the same time, it's also activating the plasmin or the plasminogen to become plasmin. Why is that? Because plasmin that's an enzyme that will start to chew off your blood clot. That will chew off the fibrin. And you want that. You want that to limit the growth of your blood clot by activating the plasminogen. So there is no wonder now one of the most important treatments for people with thrombosis when they're coming with stroke in the brain is to give them plasminogen activator, because that will activate the plasminogen into plasmin, and plasmin will start to chew off or causes dissolution of the clot, and therefore inhibits the blood clot formation. And this is very important mechanism, A, to help preventing the spread out of the blood clot, but also B is to once the bleeding stopped, the blood clot is irrelevant, and we have invited fibroblasts, and the fibroblasts now are bridging the gaps. So why do we need the blood clot anymore? Its presence can cause embolism or disseminated pieces of blood clots swimming around and lodging themselves in sensitive areas like in the lungs or the brain, and you don't want that to happen. So it's very important to eliminate it, and plasmin is a very important factor in eliminating that clot after it's formed, and preventing its existence after it's already done its function. So plasminogen activators, I want you to remember that term, and the functions of plasmin as well. With this, this is the final slide here. Things can go wrong, just like everything we studied in physiology. Things can go wrong. If you have abnormal intravascular clotting, we'll call that thrombus. And if that starts breaking up and pieces of that thrombus becomes engaged in small arteries or small blood vessels, causing embolism or emboli, and this embolism will cut off the blood supply from anything distal to that embolus. And of course that's a problem that can cause infarction of the brain, infarction of the kidney. It can cause a lot of problems if it's massive thing happening in the lung. And
We know that there are factors that can cause thromboembolism if the blood is slowly moving, whether because the person is sitting for too long time or the person is bedridden, person that has been in bed for a very long time, or has had a cast over his leg for extended period of times. Then all of that will contribute to the fact that the blood is not moving as fast as it should. If you increase the roughness of the blood vessels, then that will cause the platelets to aggregate to it. So whenever we have athero or arteriosclerosis, then you have an atherosclerosis, you will have the problem of spontaneous thrombosis happening. If you have any mechanism that will abnormally enhance the clotting mechanism, also you will start to have this thrombosis. If you have extremely high viscous blood, you will also enhance the process of thrombosis. Hemophilia is the opposite. Hemophilia is that you have excessive blood loss, and that's due to the lack of one or more of the factors. Remember the 12 clotting factors we briefly described. The more severe the type of hemophilia will be attributed to very critical, like thromboplastin or fibrinogen lack, and if you don't have that, then that's really a problem. Or prothrombin or thrombin. All of these guys can contribute to the fact that the person has hemophilia, and there are different types of hemophilia. We call them with the Roman numerical hemophilia type, X hemophilia type, whichever. That is will be counted by which type of protein is lacking. That disease used to be fatal, but now with the recombinant technology we can inject those factors back in the blood, and that saves lives. And nowadays they made even with gene therapy, they created cells, and they can take cells from the liver and put it back in the person, which will secrete to you constant amount of clotting factor, and that has helped greatly in treating those patients with this very bad disease, the hemophilia. Hemophilia, as we know, it is a hereditary disease, of course, because of the lack of one or more of those of factors. Okay, so we covered the blood chapter. We did talk about the blood elements, the red blood cells. We started with it, and then we cruised quickly through the white blood cells, because we did cover that before when we talked about the immune system. We did talk about the composition of the plasma and how important that is in many aspects. And we did talk about the platelets and the process of blood coagulation and the hemostasis. A very important chapter, and it's very, very relevant to everyday's life. We did talk about the blood types, the A and the B and the O system, but we also talked briefly about the RH, and we described the process that can go wrong in pregnant women, which we called erythroblastosis fetalis. And finally we talked about some abnormalities of blood clotting. So that concludes our chapter, the blood physiology chapter, and I hope you will find this video helpful when you are studying for your exams. And please join me for the future chapters that we will be covering together here or in our lectures together. Have a great